Hello friends, welcome back to my YouTube channel Doctor's Corner. Today we are going to discuss one of the very important topic, electrocardiogram, or also known as ECG. This ECG is also abbreviated by EKG. Okay, in some countries this ECG is abbreviated as EKG also, both are one and same. Okay, that is basically electrocardiogram. Now in today's lecture, we are going to discuss the very basic of the electrocardiogram, that is the definition of electrocardiogram, how it is produced and how it is recorded. Okay. Now, we are going to make a series of lecture on this topic right from the beginning, that is from the basic, that is the physiology, electrophysiology of the heart and the formation of the ECG waves, and we'll correlate the things with the clinical applications. Okay. So it will take many lectures for that. So we have split this topic into various parts. So today is the part 1. So I request those who have joined to our channel for the first time, so please subscribe our channel Doctor's Corner, and also press the notification button for our upcoming videos. Okay, now coming back to this topic, ECG. So what is ECG? That is the definition of the ECG or electrocardiogram. So in simple words, electrocardiogram is nothing but the graphical recording of the electrical activity of the heart. Okay, as you know, there is certain electrical activity which is present in our heart. If we represent or record that same electrical activity which is generated in our heart on a graph paper or a graphical recording, it's known as electrocardiogram. That recording is known as electrocardiogram, and the procedure of recording this, it is called as electrocardiography, and the instrument, that is the ECG machine which we use to record this, it is known as electrocardiograph. So electrocardiography is the procedure, electrocardiograph is a name of the instrument, and electrocardiogram is just but a graphical recording is known as electrocardiogram. Okay. To be more specific, what is ECG? To define, it is the summed electrical activity of all the cardiac muscles which is recorded extracellularly. That is extracellular recording of the sum of all the cardiac muscles electrical activity. Okay. During the cardiac cycle. So now coming to the very basics, we'll study this at the cellular level. So consider this as one of the cell, cardiac muscle cell. Okay, a cardiac muscle cell. So like any cells which there are 3 types of the muscle cells: skeletal muscle cell, smooth muscle cell. This is the cardiac muscle cell. This is one cell, adjacent cell, another one more cell. Okay. So I am just drawing a few cardiac muscle cells to understand this. Now before going to the main topic, electrocardiogram, that is electrical recording, how we record on a graph paper, we should know what exactly this electrical activity means. Okay, what this electrical activity means, and what is the voltage and how it is transmitted from one part of the heart to another part, and how ultimately we record this. Okay, so please give your kind concentration. We are going from basics, so it may take some time. So I think this video may extend up to 30 to 40 minutes, but it's worth to know the basics. If we know the basics, then only it's very much easy to interpret the ECG graphs later. Okay. Anyway, consider this as the cardiac muscle cell. So to know these things, you are supposed to know certain very basic terminology. One very basic terminology is RMP, that is resting membrane potential. So what do you mean by resting membrane potential? Okay, so it is the potential difference across the cell membrane at the resting condition of the cell. So I have already made one lecture on this topic, resting membrane potential. Those who don't know the details about this resting membrane potential, I request them to please go back and check on our playlist on this channel of Doctor's Corner. You will get this in general physiology playlist of resting membrane potential. Okay, so this resting membrane potential is present basically in almost all cells of the body, and specifically for this cardiac muscle, it is around minus 90 millivolt. Okay, so remember, resting membrane potential is present in all cells of our body. So what exactly this resting membrane potential means, so inside of the cell, if we see the electrical charge, it is negative as compared to the outside of the cell. How much negative? The magnitude here is minus 90 millivolt. Okay, it varies from cell to cell. For example, a neuron, it will be around minus 70 millivolt. For a smooth muscle, around minus 55 millivolt. So it will vary from cell to cell. For the cardiac muscle, it is roughly around minus 90 millivolt. Okay, so how we record a resting membrane potential? So basically it is measured by a voltmeter, or also known as cathedral oscilloscope. One of the electrode is placed on the surface of the cell membrane. Okay, suppose another electrode is also placed on the surface of the cell membrane, then no voltage difference is shown. But if one electrode is inserted inside the cell with the help of a micropipette containing an electrolyte solution, then the potential difference across the cell membrane can be determined, and at the resting condition, when the cell is not working at the resting condition, the resting membrane potential of a cardiac muscle cell is minus 90 millivolt. Okay, hope you understood what is resting membrane potential. Now, this resting, if we plot this same resting membrane potential on a graph, so on an axis, x-axis, we take the time duration in millisecond, and on y-axis, we take the voltage, that is the electrical current voltage in millivolt. Okay, so at the resting condition, it is around minus 90 millivolt. Okay, minus 90 millivolt will get a straight line, minus 90 millivolt in this graph. Okay, now if the cells are stimulated, say electrical stimulation or any other stimulation, suppose if we stimulate this cell with an electrical stimulus, okay, with an electrical stimulus, now what happened to this electrical activity of the cardiac muscle cells? So what will happen, that is an action potential is generated. Now the second important term you are supposed to know is, what do you mean by action potential? Okay, now we are going to discuss the action potential in relation to the electrophysiology of the heart. So whenever a cell is stimulated, then what happens is, there are certain ion channels which are present on the cell membrane. Okay, ion channels which are present on the cell membrane, sodium channels, potassium channels and all. So there are leaky channels. So basically the sodium channels will get opened, and sodium ions tend to enter from the exterior to the interior of the cell. Why? Because the concentration of the sodium is more in ECF as compared to the ICF. Because of the concentration gradient, it enters here, and this stimulus will open the sodium channels. Now, as the sodium ions are entering inside the cell, what do you expect the electrical activity inside the cell? So this negativity turned to become positive. Okay, because of the positive charges which is coming inside, cations which are coming inside. So from minus 90, the current will, the interior of the cell will become minus 80, then minus 70, then minus 60, so and so, until the threshold potential is reached, until a threshold potential is reached, and finally more amount of the sodium will enter through the voltage gated sodium channels. Voltage gated sodium channels, more amount of the sodium is entered. Okay, now the more amount of the sodium is entered, so the more it will the interior of the cell again will tend to become the positive. Now it will cross the plus 0, it will become plus 10, plus 20, plus 30, like that. Okay, now this phase of the change in the electrical activity of the cell, where the cell interior of the cell become positive because of the entry of the positive ions like sodium, that is voltage gated of sodium channels, this phase of the electrical activity is called as depolarization. Remember, this is very important with respect to the electrocardiogram basics, what we are going to discuss now. Depolarization at the resting condition of this cell, that is when it is minus 90 millivolt, so that is called as the polarized state of the cell. Now when the resting membrane potential is changed and the interior of the cell tend to become positive, and this phase, due to the opening of the sodium channels, it is known as depolarization. Remember, very important. Now after some time, these sodium channels get closed. Now there is opening of the calcium channels. There is opening of the calcium channels. This red color channels is the calcium channels. So this calcium channels will again lead to increase in the positivity of the cell. Simultaneously, there is also opening of the potassium channels. Okay, now with the help of this green color, what I am drawing is the potassium channels. So potassium channels, that is the potassium ions will go outside. From inside it will go outside. Why? Why means because the interior will have more amount of the potassium with respect to exterior, that is the concentration gradient. Due to the concentration gradient, potassium tend to move outside. Now calcium ions is entering inside positive, and also this potassium ion is going outside. Okay, so now almost the positive charge is entering and positive charge is leaving. So for some time this membrane potential will remain constant. Okay, this is known as plateau phase in the action potential, because slowly calcium is also entering and slowly potassium is also leaving. So there is no much decrease in the membrane potential. It will be remaining at a constant level, that is known as plateau phase. Okay, so this plateau phase is again due to opening of the calcium channels, simultaneously opening of the potassium channels also. Okay, but after some time again this calcium channel closes, but this potassium channel remains open for certain longer duration of time, and when it is kept open and the calcium channels are closed, what will happen is the more amount of the potassium is lost from interior of the cell to the exterior of the cell. So what it means, that is the positive charge is lost from inside of the cell to the outside, and ultimately this the membrane potential, if we record it with the help of this cathode ray oscilloscope, it will tend to come back to this resting membrane potential, and this phase due to the opening of the potassium channels is called as repolarization. Remember these two very important things. In our discussion we are going to use this term depolarization and repolarization. So what do you mean by depolarization? Depolarization means a positive current is formed inside the cell because of the entry of the cation, that is depolarization. So remember, depolarization means positive current. Repolarization means negative current, that is the interior of the cell tends to become negative, that is from plus 30 to again it will come back to minus 90 millivolt. So this process or this stage is called as repolarization. So in a cardiac muscle you get a plateau phase also, unlike skeletal muscle and neuron. There is just a sharp depolarization and repolarization. Anyway, we are not discussing in detail about this action potential, and it's this one. Why we are interested to is just to know about these two terms. So I hope it is clear what do you mean by depolarization and what do you mean by repolarization. So when we use this term now in our discussion with relation to ECG, remember only one important thing: depolarization means positive current, that is interior of the cell will become positive. Repolarization means, remember, negative current, that is interior of the cell will become negative. Okay, hope this is very much clear. Now what happens, this electrical current which is formed in the form of action potential. So the electrical activity of the heart when it is stimulated is nothing but the action potential. So when this action potential is formed, it is now transferred from this membrane, then this action potential is shifted to the neighboring cell. How this action potential is stimulated or transferred to the neighboring cell is with the help of gap junctions. Okay, there are certain gap junctions which are there in between the two adjacent myocardial cells. So basically they are filled with some certain amount of the fluid, and this sodium which is entering inside the cell because of the stimulation of this particular cell, this sodium ions tend to move inside the another cell, okay, through this gap junction. So when they move inside the another cell due to the gap junction, so here the resting membrane potential was again minus 90 millivolt. So again this sodium ions will again triggers the threshold potential, and again this series of events will start, opening of the sodium channels, potassium and calcium channels, and ultimately there is an action potential generated in this cell also. Again same thing. So when this cells are stimulated through the gap junctions, again this sodium channels, through the gap junction sodium ions will go to the adjacent cells, and this is how the action potential, that is the flow of current from one cell, it will passes, that is depolarization and repolarization current passes from one myocardial cell to another cell and another cell. Okay, why I am discussing this concept so in detail? To understand this thing, that is electrocardiogram. I have not yet started. What does it mean? Okay, so to understand this you are supposed to know these basic things. Okay, I hope this is very much clear. What is resting membrane potential? What is action potential? Okay, resting membrane potential is the potential difference across the cell membrane in the resting condition, and it is minus 90 millivolt. Action potential is when the same cell is stimulated, the series of electrical events what is recorded here, that is depolarization and repolarization, that is known as action potential, and the action potential is nothing but that the electrical current which is passing from one cell to another cell. Okay, now this depolarization and repolarization, these phases, that is this electric current which is moving from one cell to another cell, that is inside the myocardial cells, is recorded extracellularly on from the surface of the skin. Okay, you know that our body is a very good volume conductor, that is it can transmit the electrical signals. It's a very good conductor of electricity. So this heart, this electrical activity, basically it is in the myocardial cell, that is the cardiac muscle cells. So this electrical activity is transmitted through the body fluids to the surface of our body, that is to the skin. So now if we put certain electrodes on the surface of our body and record the potential difference between the 2 points, we are ultimately recording the cardiac muscle activity, and this how this cardiac activity is represented, that is known as electrocardiogram. That is the graphical representation of the electrical activity of the heart. Now let's see let's see how this ECG recording is taken place, and how this electrical activity is recorded, and what are the different waves we see on an electrocardiogram. Okay, so hope this point is very much clear. If you have any doubts, please type in the comment section. I'll be pleased to answer and discuss the various doubts raised by you people. Okay, so for it's already around 15 minutes. So again, next 10 to 15 minutes we are going to discuss the very basic concept in this particular lecture. Later in the next lectures we'll discuss about the further concepts of the electrocardiogram. Okay, now come back to the heart muscle, the topic proper, that is electrocardiogram, ECG, how it is recorded and what are the waves in the ECG. Again, to understand this, again we have to go back to the basics here. So I am drawing a very rough diagram, a line diagram of the heart. Okay, so as you all know that heart is made up of 4 chambers. Okay, what are that 4 chambers? 2 atrium. Okay, right atrium and left atrium. Okay, this is right atrium, this is left atrium. Okay, and 2 ventricles. 2 ventricles, it is the right ventricle and left ventricle. Okay, so left ventricle generally is thicker as compared to the right ventricle. Okay, this is the cardiac muscle. I am doing just a very basic thing, that is the line diagram of a heart. Now, how the electrical activity, that is this depolarization, which is a positive current, and repolarization, which is the negative current, is transmitted in the heart, we are going to discuss. So the main, as you know, the main tissue which will originate this conduction of the electrical activity, it is the SA node. Okay, which is usually present in the right atrium. Okay, right atrium, that is the SA node. So from this SA node, the impulse is generated and it is transmitted to the different parts of the heart, and that electrical activity is recorded extracellularly. Okay, so to understand these things, again we have to go back to certain basics. We are supposed to know certain phenomena. Again, suppose if I take a small patch of this myocardium. Okay, you know epicardium, myocardium and endocardium. So the myocardium, that is the cardiac muscle. If we take a small patch of a myocardium here, suppose if I take one small patch of a myocardium here now, and put one electrode, that is one negative electrode here on one side, we will mark it as a point A, and one positive electrode will mark it as point B. Okay, minus negative electrode and positive electrode, and both this electrode is connected to a galvanometer. Okay, galvanometer, which will record the change in the electrical activity. So negative electrode here and positive electrode here. Okay, to the piece of a cardiac muscle. Now in this galvanometer there will be certain recording device. A pointer will be there which show any deflections. Okay, generally at the resting condition. Okay, so this is a cardiac muscle, it consists of the various cardiac muscle cells. Okay, consider this is a piece of a cardiac muscle which has so many cardiac muscle cells. So remember this very basic concepts what I am going to tell you, because if you understand this, you can understand why there will be so many different waves in an ECG and what is the cause of the waves, so many waves of the ECG. Okay, now to this cardiac muscle, at the resting condition, when there is no electrical activity in this cardiac muscle, if we see the recording in a galvanometer, which will record the electrical differences between the 2 points, so it will be 0. You will get a very straight line, that is, there will be no deflection of the needle of the galvanometer. Now, when there is a depolarization, so what do you mean by depolarization? Depolarization means positive current, okay. So when you stimulate one cell here, so that is positive, interior will become positive. Remember, just now we have discussed depolarization means positive current, depolarization means negative current. So interior of the cell becomes positive, and this positivity is transmitted, depolarization is transmitted to the adjacent cells through the gap junctions, okay. So this wave of depolarization, suppose if we stimulate here, is transmitted in this direction, okay, in this direction, that is from this negative electrode to positive electrode, that is from point A to point B. So this depolarization wave goes from this one cell to another cell in this direction. So when the positive current means depolarization current, is moving towards the positive electrode, then there is a deflection in this galvanometer, and this deflection in the galvanometer is towards the positive, and it will move up, okay, it will move up. Whenever, remember, positive current moves towards the positive electrode, then the deflection is shown as a positive sign, that is, deflection will be moving up. This galvanometer, this pointer will be moved up. Now, similarly, suppose instead of stimulating on this side, that is point A, if we stimulate this cardiac tissue on this side, that is point B. So here again the depolarization will start from here, and it will, the flow of the current will be from B to A, that is from this positive electrode to the negative electrode. So when this positive current is flowing from positive electrode to the negative electrode, then the deflection in the galvanometer is shown as negative, that is, this electrode, this pointer will move down, okay. So it will move down. So remember, if positive current moves to the positive electrode, it means same charge moves toward the same electrode, then there will be positive deflection. If one electrical charge is moving towards the opposite charge, for example, positive charge is moving towards the negative electrode, then it will show negative deflection, okay. This is one rule number 1. Conversely, suppose if the negative charges, that is repolarization is negative current, depolarization positive current. Suppose there is a repolarization of this tissue. If the repolarization starts here and the negative current passes towards the negative electrode, what will be the deflection here? So same charge, if suppose there is a negative current which is passing in this direction towards the negative electrode, then again there will be positive deflection. So same charge, when it is going to the similar electrode, it is showing a positive deflection. If it is going to the opposite electrode, it is showing negative electrode. One more thing, when there is a complete depolarization has taken place, or complete repolarization has taken place, at that time there is, when it is completely depolarized, again there will be no electrical activity, that will be electrical silence will be seen, okay. So remember these basic things, will help us to understand the very basic concept of the ECG. Now coming back to this heart muscle, how the electrical activity passes through this heart muscles in a heart, and how waves are generated in ECG, okay. Now consider this as a human being. So again I'll just draw a very rough diagram. This is suppose a human being, okay. Heart is not supposed to be so big, but just for the sake of understanding, this is where I'm shown heart as a big, as trunk. So this is face here. So this is the left arm, left upper limb, and this is right arm, okay. Here there is left leg, right leg, okay. Just hypothetically imagine that this is a human being. Inside this is a heart. Heart is definitely a smaller one in the precordium as compared to the trunk, but here I am shown big to understand the concept here. So there is a left limb, left upper limb, right upper limb, left lower limb, right lower limb, okay. Now suppose if we keep one electrode on the right upper limb, that is on the right arm. Okay, if we keep this one electrode, one positive electrode on the right arm and one negative electrode on the left leg. Okay, for the sake of convenience, generally when we record an ECG, there are different types of leads which we are going to discuss after this concept. So that leads or electrodes are placed suppose here. So instead of here, we can even place up to the distal part of this forearm, because is as good as recording the electric current here. Okay, right arm. Instead of this point, we can keep it to the lower part of the left lower limb. Okay, so anyhow, for this discussion, remember here there is a, suppose we'll keep here negative electrode. Okay, we are keeping here one negative electrode and positive electrode here on the left foot. So right arm there is a negative electrode and on the left foot we keep a positive electrode. Okay, generally this type of arrangement is there in the bipolar limb lead too. Okay, we'll discuss what is this limb leads, then chest leads, unipolar leads, bipolar leads, augmented leads. So before that, what are the waves of the ECG and how it is produced, we are going to discuss now. So this negative electrode is now connected to a galvanometer. Okay, galvanometer which will record the potential differences. Okay, galvanometer. So it has a pointer and over this pointer there is certain ink is placed, okay, to write the things. So negative electrode is on the right arm and positive electrode is on the left leg. So positive electrode is placed on the left leg and negative on the right arm. Now this galvanometer is basically the ECG machine, okay, which will record the electrical activity of the heart by placing the electrodes on the surface of our body. Okay, we are placed two electrodes on the surface of our body. It will record the electrical activity in between these two electrodes. Okay, and it is recorded by a galvanometer. So basically this ECG machine is a galvanometer where with a pointer, underline this pointer, there is one paper, ECG roll is kept. Okay, ECG paper which is fitted by a motor and it is constantly rotating or evolving with a particular speed. Okay, I will just discuss what is speed later, how much speed is kept and all, what are the markings on an ECG paper and all, we will discuss later. Timing, remember, just there is an ECG paper and there is a pointer over this. So whenever there is a deflection and it is moving, the deflection is shown as certain waves. And that how that waves are generated, we are going to discuss now. So remember, first thing is this SA node. So the electrical activity of the heart, this SA node will stimulate this atrial muscle. So atrial muscle, and it causes the depolarization of this atrial muscle. Remember, in between the atria and the ventricles, there is a fibrous tissue, okay, which acts as an insulator. We are going to discuss this thing again now. Just timing, remember, there is a fibrous tissue and this atrial muscle, it gets depolarized, that is this SA node is stimulated and it causes the depolarization of the cardiac muscle cells, that is the atrial muscle cell. Depolarization means positive current, and the current will move in the downward and towards the left, the direction of the current. Now one more very important thing, concept of vector. So a vector is basically an arrow which will tell the magnitude and direction of a force. So here the force is the electromagnetic force and the direction of the arrow shows the direction of the force, that is the direction of the current here. So we can record this electrical activity in the form of a vector. So vector will show this electrical force direction and the length of this arrow will show the magnitude of the force. Okay, if the electrical activity is very small, the magnitude of this arrow will be less. If the electrical activity is of more strength, then the length of the arrow will be more, and the direction will point toward the direction of the flow of the current. So here from this SA node, and when it is stimulated, it causes the depolarization of the atrial muscle cell. Now remember, just now we have discussed what is depolarization. The interior of the cell will become positive and it will causes the depolarization of this neighboring cell. So both left and right atrium, because this SA node is situated in the right atrium, so this depolarization waves goes towards the left atrium muscle also. So this atrial muscle, that is the positive current, depolarization means positive current. So this positive current moves to these atrial muscle. Now imagine the same thing. Here there is a positive electrode, here there is a positive electrode, and here there is a negative electrode. So when the positive current is moving towards the positive electrode, what deflection you will expect? Positive current is depolarization in the positive current, it is moving towards the positive electrode. So here the recording will be positive. So at the resting condition, when the SA node is not given any signal, then there will be straight line. But when it is stimulated, it will cause depolarization of this atrial muscle, and this depolarization of the atrial muscle causes a positive deflection here, and this paper underneath this writing pointer, so it will be moving, and we get a one small wave like this due to the depolarization of the atrial, also known as atrial depolarization, and this wave is known as P wave. Okay, P wave. So what does this P wave represent? It represents P wave on ECG. Now this is the ECG, that is electrocardiogram, that is the graphical representation of the electrical activity of the heart. Now we are correlating the electrical activity of the heart with the ECG waves. Okay, how ECG waves are recorded, we will go in further details later. Timing, just we are concentrating how this electrical activity is recorded on ECG paper. So this electrical activity of the atrial muscle, that is depolarization of the atrial muscle, is represented as P wave. Okay, why positive deflection? Because the positive current is going towards the positive electrode. Okay, now as I said, there will be a fibrous tissue between the atrial musculature and the ventricular musculature. So this acts as an insulator. So this positive depolarization current won't go to the ventricular muscle. There is only one way for this depolarization current to pass from the atria to ventricle, and it is through one more specialized conducting tissue in this heart, that is known as AV node or atrioventricular node. Okay, atrioventricular node. So remember, the first electrical activity is the atrial depolarization. Okay, I'll mark this arrow, that is the vector, and this is the first electrical event which causes the P wave. Now this electrical signal now passes to the AV node, also known as atrioventricular node. Now the conduction in this AV node is very much slow. Okay, very much slow, just 0.05 meters per second. Okay, very slow. Okay, conduction speed is very slow as compared to the Purkinje fibers. Purkinje fibers here we'll discuss now later. So that Purkinje fibers, the speed of the electrical current will be around 4 meters per second, but here it's just around 0.05 meters per second. It's very slow. So electrical activity in this AV node, it is very slow. The current, basically the flow of the current is slowed down here. Okay, it will take much time. Okay, and the magnitude of the current is also of a very minimal strength. Okay, that is of low strength. So this is the second electrical activity which is there in the heart. So we'll draw again one more vector. It is again in the same current is going down through this AV node, but it is of such a minimal strength that this current is not detected by the ECG machine. Okay, the depolarization wave which is passing through this AV node is of very low magnitude. The strength of the current is very low magnitude and is very slow, that it is not detected by the galvanometer or the ECG machine here. So it will appear as a straight line, as if representing the isoelectric line, and that is the time of electrical silence of the heart when the current is flowing through the AV node. Okay, and this straight line is known as isoelectric line, and it is called as PR segment. Okay, this line is called as PR segment. Okay, we'll discuss about this segment and all later. Timing, remember, there is one isoelectric line when the current flows through this AV node. Okay, now what is the significance and all we discuss later, but here again I would like to tell it. So why there is the flow of current is slowed down through this AV node? Why means, because when there is a depolarization occurring in the atrial muscle, it is followed by the mechanical event of the cardiac cycle, that is atrial systole. When there is a depolarization, the atria will contract. Okay, now if at the same time if the ventricular muscle is also depolarized, then ventricular muscle will also contract at the same time. But when the atria is contracting, the flow of the blood through these walls is from the atria to the ventricles. If simultaneously even the ventricles are contracting, that the blood would not get accumulated in the ventricles, okay. That's why the current is slowed down. That is, when the atria is contracting, the ventricles has to relax. So when it is relaxing, then only the ventricles will contract, okay. There should be some delay. So that delay, it is provided for the current by this AV node, okay. The current second electrical event is the passes through the AV node, and this is very minimal, not detected, and it will appear as an isoelectric line, also known as PR segment. There are certain wave segment and intervals we'll discuss later. Now, from this AV node, the conducting tissue, it will go to the bundle office and the left branch and the right branch, and ultimately through the Purkinje fibers to whole of the ventricular musculature. That is the conducting system of the, this is the conducting system of the, this conducting system, that is this Purkinje cells and this AV node cells are basically, they are modified myocardium, that is similar to the myocardium but modified myocardium, specialized myocardium, okay, specialized myocardium. And this here, the conduction of from this Purkinje cells is more, as I already said, it is 4 meters per second as compared to just 0.05 meters per second to the AV node. Why means, again here the resting membrane potential of the AV node is minus 60 millivolt. Here resting membrane potential of the Purkinje cells is minus 90 millivolt, and the size of the cell of the Purkinjes are more, and the size of the cells of the AV nodes is smaller, okay. There are many differences, we'll discuss later. Timing, remember, the conduction speed is more here and it is less here. Now, the depolarization, wave of depolarization through the AV node, ultimately it will reaches the interventricular septum. Now the ventricles are depolarized in 3 parts. First, the interventricular septum is depolarized. So more specifically, the left bundle branch here will get, first it will cause depolarization of the interventricular septum, and the direction of this depolarization, that is from the left, it will go towards the right and the upside, okay. So this depolarization current, if we draw a vector here representing this electrical current of the interventricular septum, that is depolarization, the current is going, and it is, the magnitude is very small because it is just depolarizing the cells of the interventricular septum here. So this interventricular septum, and the direction of the current is moving upwards and towards the right, because the left branch is first stimulated, and it will stimulate and pass the depolarization current here. Now this depolarization current is the positive current, and this is third electrical event. First was this atrial depolarization, second was passing through the AV node, third electrical event. So this third electrical event, that is the depolarization of the interventricular septum. Here the positive current is moving away from the positive electrode. So what type of deflection you expect? Yes. So the deflection here is the negative one. So the deflection of this recording device is negative here. The pointer will point down negative, and it will produce one wave that is known as Q wave, a negative wave. So this negative Q wave, it is because of the depolarization of the interventricular septum. Now after this Q wave is depolarized, then the wave of depolarization spreads to the whole of this ventricular musculature from this endocardium to the epicardium, that is outside is pericardium, inside is endocardium. So from this inner layer of the myocardium, the wave of depolarization now passes to the outside, both in the right ventricle and also in the left ventricle, okay, from the inside. That is the major ventricular depolarization takes place. That is the major portion of the ventricles get depolarized here. Now if we draw a vector of this fourth electrical event, that is the depolarization of both the ventricles. So right ventricle depolarizes here, the direction is here, left ventricle depolarizes there. But if we take the sum of this whole of this ventricle, major portion of the ventricle, the vector is represented by a larger vector, because the ventricular musculature, it is more as compared to the atrial musculature. So the vector size length is more, and the again direction, it is pointed towards the left and downwards. Why left? Because the current here, the left ventricle is bit thicker as compared to the right ventricle. So this is the fourth electrical activity. Now this positive current is moving towards the positive electrode. It will leads to a positive deflection, and this is represented by one bigger wave here and an ECG, and this wave, it is known as R wave, okay. So first one is the P wave, that is atrial depolarization, then Q wave, the interventricular septum depolarization, and finally this R wave, a bigger wave because of this bigger vector of the depolarization of both this ventricular muscle. Now finally the basal portion of these ventricles are depolarized. Basal portion of this ventricle depolarized. So ventricle depolarized in 3 phases. First, interventricular septum, the direction of the current is upwards. Then the major portion of the ventricles, that is the direction is downwards. Then the basal portion of the ventricles, again the basal portion of the ventricle, it gets depolarized last, and the direction again here is upwards and towards right. So again it's a small muscles, that is small amount of the cells are depolarized here. So the length of the vector is small and the direction of the current is upwards. So here if the positive current is moving towards the negative electrodes, then again the deflection is shown as negative, and this negative wave is known as S wave. So whole of this QR and S wave is called as QRS complex. So this QRS complex, it represents the ventricular depolarization. Okay, it represents the ventricular depolarization. So what does this P wave represent? It represents the atrial depolarization. QRS complex represents the ventricular depolarization, and this PR segment represents the passage of current through the AV node. Now when this ventricular is depolarizing, simultaneously this atria will cause repolarization, that is the negative current again passes, but this negative current is not detected by the ECG, it is masked by the ventricular depolarization wave, that is which is a stronger vector, that is QRS complex. That is during this QRS complex only the atrial repolarization takes place, but that is not marked by another separate wave because it is masked by this ventricular depolarization wave. So atrial repolarization wave is not seen, it is masked by this ventricular depolarization wave. Now when the whole of this ventricular muscle is depolarized, so for some time before the repolarization starts, when the whole of this ventricular muscle is depolarized, so for some duration there will be one straight line, that is an isoelectric line is seen. Okay, same like this PR segment, that is known as ST segment. Okay, it is known as ST segment. So what does this ST segment signifies here is, so whole of this myocardial muscle, ventricular myocardial muscle is depolarized, and there is no net movement of the charges when there is whole of this muscle gets depolarized. So at that point there is no movement of the pointer here, and it will form an isoelectric line that is known as ST segment. Again further we will discuss what are the different segments and intervals, but timing remember it is known as ST segment. Now after that sometime the phase of repolarization starts, that is that plateau phase is there. After that again repolarization start, depolarization over, now repolarization of the ventricle start. Repolarization means negative current, so negative current will flow now. So remember one rule is there, the part of the ventricle which is depolarized last is the first to repolarize. Okay, so suppose here the outer portion, that is the outer portion of this ventricular muscle, it's the last to depolarize. Now that same portion is the first to repolarize. So repolarization is the negative current. So the negative current repolarization, that is the negative current will be moving from this direction, that is from the outward to inward. So negative current, that is the repolarization current, will be moving from outward to inward, that is, and remember this negative current again if we put it in a vector, so this electrical event, that is the last one, that is the fifth electrical event, we put it on a vector here on this black color, with this black color. So direction of the current is upwards. Okay, upwards and towards the right side, towards this electrode, negative electrode. So when this negative current is moving towards the negative electrode, the deflection is positive. Why? Because when the like charges move towards the similar electrode, it will make a positive deflection. So we will see a positive deflection here, and that positive deflection, it is termed as T wave. So what does T wave signifies? T wave signifies the ventricular repolarization. Atrial repolarization is not seen because it is masked by the QRS complex wave, because it is very strong. Now after fully, when the ventricles are depolarized, the repolarization wave, that is the negative current, it is passed in the opposite direction, that is it will pass to the negative electrode. Negative current passing towards the negative electrode, it will cause a positive reflection, you get this one. So basically, to summarize, we get this waves on an ECG, it is P wave, QRS complex, and T wave. P wave represents atrial depolarization, QRS complex represents ventricular depolarization, and T waves represent ventricular repolarization. Okay. So again, in the next video, we'll discuss the various waves in detail and the recordings on an ECG paper. So please subscribe and like our channel. Thank you.